So I think you received the email. So that's from me. We are very lucky to have our visitors from the UK. So these are actually a collaborative work among UPM, UK, and Shanghai. But our collaborator from Shanghai they, cannot make it because of the situation in China. But they are joining us using the Zoom room now. So it's being done hybrid. So so I would like to introduce our speakers today. So we have Professor Andrew Fleming. Can you see? Dr. Andrew Fleming and Dr. Jennifer Sloan. So briefly about our speaker. So Professor Fleming, he obtained his first degree from Cambridge in natural sciences, and then did his PhD in Scenseme University at Cambridge in plant science, and then it's followed by the second doctorate, habilitation at the University of Bern. He's currently the Director of Research and Innovation at Faculty of Science, University of Sheffield. And he's got a number of grants. That's many grants. So I'll just mention. So the grant that we have now, it's sponsored by the Royal Society. It's from the UK. And so Professor Fleming is also the advisory board for neophytologist, advisor to the Commonwealth Scholarship Committee, and also the Gatsby Plant Science mentor, just to name a few. So, and he's been actively organizing a few meetings in the past involving rice. So the recent one in 2020, it was UK Rice Early Career Research Symposium, which was held in Sheffield. And actually, Professor Fleming came here before in 2018. It was in this room, but the projector was just fine. So apology in advance for the quality of the projector. I guess it's been a while since we last had our physical event like this. So probably it's a bit rusty now. Yeah. And then we have Dr. Jennifer Sloan. So Dr. Sloan, so she grew up in Isle of Wight in the south of UK. It's a little island there. I haven't been there, but I know. Is it warm there?
A little bit warm.
It's closer to France, right? So, yeah. So she did her first degree in Sheffield, studied plant science, and then she continued doing similar plant science for her PhD under Andy Fleming. And then she continued doing the postdoc with Simon McQueen Mason at University of York on using hemp, the cannabis sativa, as a biofuel. In 2014, so for a few years actually, she was a technician working with Julie Gray at the University of Sheffield on stomata function in Arabidopsis. And during this time, she had 2 lovely daughters. Right, yeah. I actually have known both of them for quite some time. So it's more than 10 years now. So we've been working together. So you know, we just good scientists get together and creating interesting stuff. All right. And currently, since 2018 and to date, doing the postdoctoral with Andy Fleming, of course, and then joined work with me and Shanghai, working on rice leaf development, modeling photosynthesis, rice plant, tillering, and see what are the effects of CO2 have on rice. Okay, so without further ado, I would like to invite our speaker and have a talk. You.
Deeply warm welcome that we've been provided, and okay, I have two microphones, so that might. Which one shall I use? Both. Okay, fine. Okay, so again, thanks so much to Armin and all his colleagues for the extremely warm welcome that we've received. It arrived, I think, last Saturday, and we've been traveling up to see Mardi and Perit and other parts of Malaysia, and it's a great trip. Also, to start by saying apologies for the slide quality is not perhaps exactly as we would like, but I understand also this is about the first face-to-face meeting that you've had in a long time, so I think it's really great that this happens. I mean, in Sheffield as well, in the UK, everyone's been suffering from the pandemic, and we've learned a lot about using Zoom and various hybrid meetings, but we find that still actually getting face-to-face with people and talking with people, nothing really beats that. So it's really great to have the invitation and to have the opportunity to be here. I'm standing here giving a talk, but actually it's going to be sort of two talks. The first session is going to be pretty much about fundamental science that we've been doing. And then there'll be, I think, a short break. People can talk, get some coffee and tea. And then in the second part, we'll move on to looking at the effort we've been making to take that fundamental science from, if you like, the laboratory or the glasshouse into the field. In the talk, it's really a two-hander. So I'll be starting, but then at some point, I'll be handing over to Jen Sloan, who's the one who's really done most of the real work. I mean, I'm just a prof. I just write stuff and send emails. And again, I'll be talking about some of the work that's been done by Armin and his colleagues, and also a lot of the work has been done by Jingran Zhu in Shanghai and Yi Zhao, who can't be here, but they're hopefully online. So there's quite a lot of computational biology work, certainly in the first talk, and that's all been done by our colleagues in Shanghai. Okay, so that's sort of an outline of what we're going to be talking about over the next, well, hour and a half, 2 hours. Let's see how it goes. And what I wanted to do, first of all, is just to say something about where we work in the UK. And I think this first slide probably doesn't look so great, but on the, let's see if I can get a laser pointer up. Good, laser pointer should be, okay, I'll use my arrow. So hopefully you can still see this is the outline of the United Kingdom, which is normally a wet, cold and drizzly island in Europe. But this week it got to 40 degrees. So it was actually hotter in Sheffield than it was in Kuala Lumpur, which on the one hand is funny. At the one other time it's actually quite scary, because certainly when I was growing up as a child in the UK, the idea of the temperature getting above 30 degrees was just really special. I mean, I think just very rarely happened. And I think if we need evidence that climate change is a real thing, then certainly the temperatures we've reached this week in the UK indicate that some our climate. And that's part of the impetus for the work that I'm going to be talking about today, is that obviously we depend for our lives on our crops. And our crops have been bred and selected over the last 10, 20, 30 years for normal climates. And we have to be very aware and think about how are our crops going to cope in the next 10, 20, 30 years with the changing climate, not just temperature, but also changes of waterfall, intensity of waterfall, those sort of factors. So perhaps first of all, a bit more about Sheffield itself. So if you want to know where it is, again, I'll find my pointer. So the red arrow points to Sheffield up here. If you follow football, then Manchester and Liverpool are on this side of the island. I'm afraid Sheffield's not doing so well in the football stakes, but we're about an hour away from Manchester and about 2 hours from London, which is down here. In terms of the UK, it's quite a large university. You've got about 20,000 from a school of bioscience, which is about hundreds research groups, all in one very large building, very complicated building. And it covers the whole spectrum of fundamental science. So it's not just plant science, it involves microbial science, molecular biology, cell biology, evolution, ecology, developmental biology, biomedical research, so the whole spectrum of modern bioscience research. And the group that we're in is down here, that's the plants, photosynthesis and soil group, which is itself already quite a large grouping. And I'll go through that very briefly next slide. In terms of how we do as a science department, so every 7 years, the UK universities go through a scheme called the Research Excellence Framework, where every department is sort of ranked and compared with every other department in the UK. And we're very happy that the most recent one, the results were just published in 2000 well, just last year. We came 4th in the UK. So we actually are pretty competitive in relationship to other biology departments in the UK. And our strength is probably in our breadth. We are perhaps not world leading in any particular sector, but across the whole breadth of biology, I think we're pretty strong. And that's exemplified by our plant science grouping. And again, I know that this slide you're not going to be able to read, because even with a good slide, it was difficult to read. This is just going to be a blur, I suspect. But don't worry, I'm not expecting anyone to read all the names here. This is just giving an indication of the breadth of plant science that we do. So ranging from people who work on the hardcore molecular biology. So these guys here, like Neil Hunter, they're studying what happens to the light energy in the first nanoseconds of it being absorbed by chlorophyll in a plant, over to, for example, people like David Bearding on this side, who are looking at how plants influence the global climate, and also doing some quite fancy things on how we can use plants and microbes to mitigate some of the effects of climate change. And what we've done over the years is to try and build up a whole stream of people who populate the entire range of plant science, from whole global biology ecosystem level down through organ and plant biology, right down to like hardcore structural biology, what individual molecules or even individual atoms or electrons are doing within the plant photosynthetic system. My group is up here on the left-hand side. So we're sort of, I'm a molecular biologist. I'm not an agronomist. I'm not a plant breeder. That's not my area. But I'm interested in using the work that we do in applications. And to do that, then you need to link up with people who do understand about those things. And again, that's part of the motivation for our collaboration that we're having with Armin. So again, a bit more, very briefly, an overview of the sort of topics that are covered within the research in the group. We cover probably too much. A long-term interest is in the whole process of leaf development. So leaves, the primary structures which capture light and absorb the light energy and use that energy to fix CO2 into sugar, so the food that we eat. Understanding how that structure develops from a few cells into a much larger organ with all the different specialized functions that those cells have. And then as one specialized group of cells that you find on the surface of leaves are the stomata, so the micropores that open and close and allow CO2 in, but also allow water out. And through a very fruitful collaboration with Judy Gray, another colleague in the department, we've got into trying to understand how these cells actually work as they do. And then a third element of our research is to look at meristems. These are groups of stem cells in plants, and trying to understand how those stem cells continue to generate cells throughout the whole lifetime of the plant, and then how those stem cells or their derivatives of those stem cells become organized into structures that we recognize as an intact plant. So the general areas of molecular cell biology was called developmental biology, how things grow and develop. the impetus for the particular work that I'm going to be talking about, or Jen and I are going to be talking about today, comes from a funding opportunity which was started by the UK government several years ago now, and it's called the Global Challenges Research Fund. And this is a great scheme funded by the UK, which recognized that there are a lot of problems in the world, and that science can and has to play a key role in addressing and solving many of those problems. I'm sure many of you are aware of the Sustainable Development Goals developed by the UN, and again, it's a bit blurry here, but I'm sure you're aware of all the challenges that we have in terms of climate change, food sustainability, health. And so what the UK government wanted to do, or to say, was that this is not something that the UK is going to solve on its own. That's not going to happen. But the UK can maybe link up with researchers, especially in the global south, where a lot of these problems are most acute, and together perhaps start developing potential solutions for some of those challenges, and at the same time allowing the exchange of skills and knowledge between groups around the globe. So, and obviously one of the global challenges is food. Is how are we going to generate sufficient nutritious food for the growing population, or even the population that we have now? And very clear, as soon as you get into those areas, then rice as a fundamental source of food for the planet is an obvious target. So we put in a grant application. This was a joint application between ourselves in Sheffield, Armin and colleagues here at UPM, and then, as I say, colleagues in Shanghai. And it's funded by actually the Royal Society. So this is government money, but it was channeled out to the research community via various routes. And the route that we received was via the Royal Society, which I'm sure you're aware is a learned society based in the UK, lots of clever people, and they're very clever because they decided to give us money. Okay, so moving on to the actual project that we've been working on now for a number of years. The title is a modeling approach to investigating rice leaf anatomy and photosynthesis. So what does that mean, and why is it important? So I'll spend a few minutes just describing, if you like, the background to the project, and then going on to report on some of the results that we've been able to achieve. So first of all, photosynthesis. And again, this is, I know we've got quite a wide range of people in the audience, so I'll just remind people, photosynthesis is the key metabolic process that maintains life on the planet, not just us, but virtually everything else. And it requires a number of elements, in that you have carbon dioxide, CO2, in the atmosphere that is taken into the plant. And then at the same time, the plant has the ability to absorb light energy, and it takes that light energy, converts it into chemical energy and something called reducing power. And that is used to fix the CO2, the gas that's all around us, into solid sugar. Now that happens in things called chloroplasts. So these are small organelles that are the green bits that you see in plant cells. But obviously, those little organelles are all located in individual cells, and those cells are all located in a larger organ that you recognize as a leaf. So what we set out to do was to ask the question, can we simulate, capture that process in a computational model? Why would anyone want to do this? If you think about, I'm an experimental biologist, so my background is very much, you go into the lab, you set up an experiment, you run the experiment, that takes time, you get the results, you analyze the results, then you do another experiment, and that's all great fun and fantastic, but it takes time. And so there's quite a push in the whole area of bioscience, is that because of the tremendous increase in ability that we have for using computing power to capture or simulate living systems, if you can capture a system in a computational model, then you can start to interrogate that model, change different parameters and explore what's called parameter space to try and find out if you wanted to improve that system, what would you tweak, what would you change? So, and obviously, once you've got those ideas and you've got a model that works, then using computational power, you can run many hundreds or potentially thousands of experiments in silico really quickly, most of which may be rubbish, but which hopefully some of them will identify things that potentially could be useful in the real world. And then you can take the outputs of that computational model, go to the folks in the lab and say, these are the experiments that you should be doing, right? To test a particular hypothesis or to engineer a particular trait that you might think would improve that living thing or that leaf. So creating computational models is, first of all, it's scientifically challenging. So it has a certain academic interest. I mean, to what extent can you capture a complicated living system in a computational model? And those models themselves can provide insights because things like photosynthesis are incredibly complicated. And there's no way that our brains can actually understand or capture all the information at one time and predict what happens. Because very often systems work in much more complicated ways than our brains can conceive. And then I say the second thing is that once you've got that model, you can start to explore it and to ask questions. Okay, what exactly is the role of how many? Is it good to have big cells or small cells in a leaf? Is it important to have particular types of cells in particular regions? Or are there particular metabolic pathways that we would want to tweak to maybe make photosynthesis better? Because one thing, again, that the academic community has identified for many, many years now is that although photosynthesis is perhaps the most important metabolic pathway on the planet, it's actually one of the lousiest. There are many, many elements of photosynthesis that if you study, you see that's really inefficient. Now, obviously, evolution is very effective, it works, but from an engineering point of view, it just seems that there are elements of the system that are not very efficient and that therefore could be improved. So again, the final sort of goal, if you like, is that once you've created these computational models, can you use them to more quickly identify traits that perhaps we can then go to geneticists or plant breeders and say, okay, our model says if you were to change or alter that particular trait, that plant would perform better. And the key word here is climate ready. And that sort of comes back to what I mentioned before about the fact that this week it was 40 degrees in my home city of Sheffield. And Sheffield should not be at 40 degrees. There's no way. That's just so abnormal. And it means, can we use these sort of models to help guide us to improve our crops so that in 10, 20 years' time, when those sort of temperatures are probably going to be much more common, they still will either survive and still generate some sort of yield. So the overall goal is to create a model of photosynthesis and then use that to find out, can we get information that we can use to inform breeders to say, these are the traits that you should be looking at or engineering into your crops so that they're better able to withstand changing climate. Okay, so that's the basic background of the context of the project. What I'm going to do now is to move into some of the data, and again, some of the data is slightly complicated, so don't panic at any moment in time. If you understand the detail, that's fine. If you don't understand the detail, don't worry. Hopefully, I can get across the main aims of what we're trying to do. Okay, so what we have here on the bottom is what is going to be our in silico model of a leaf. Now, of course, it doesn't really look like a leaf, but again, that's not the point of a modeling exercise. The point of a modeling exercise is to, can you capture the essence, the basics, the fundamentals of any system so that it captures an element of how the system works in reality. So that's the first challenge that you have. So obviously in this computer leaf here, what you've got is the outside of the leaf, and then inside the leaf, these little green balls, these are going to be the individual cells. And the aim is that each of those cells is going to perform photosynthesis in the computer. And then we're going to ask the question, when we run the model, do the outputs of the system in terms of carbon assimilation effectively capture the real life situation of what we measure when we go out into the field or the glasshouse of how the plant works? So this involved actually a lot of work, because you have to do what's called parameterize the model. You have to get the data to set the parameters of the size of all the elements, the structure of the elements, and also the metabolism of photosynthesis within each of these virtual cells. So let's see if I can get the movie to run, and hopefully it will. So what you have here is, again, this is the outside of the leaf, and we take a lot of images of the leaf to set that. There's a particular group of cells called the bundle sheath cells. We use data there to set those parameters, and then we have particular cells, the mesophyll cells, which we measure and use that to set the parameters of the actual model. Within each cell, they're put together, and within the leaf you have air spaces, and we can estimate the airspace by using something called CT scanning. So that gives you the ratio of solid tissue to airspace within a leaf, which is shown here schematically. And then finally, we go deep down at a subcellular level using electron microscopy to get data on, for example, the amount of exposed surface area of each of these cells, the amount of basically chlorophyll and the volume of the plastids in each of these cells. And then also the final element, which is not really shown here, which is the thickness of the walls around each of these cells, because photosynthesis is a diffusional process. You have CO2 diffusing across boundaries, and therefore the thickness of those boundaries, the amount of surface areas of those boundaries will, by physical laws, have a role in limiting or controlling the flux of CO2 into the system. So again, that went rather quickly. So but the bottom line is we took a lot of images at different levels of scale, from whole leaf to cellular to subcellular, and then use those data to parameterize your structural model. Now, we were interested in looking at how rice plants might respond to climate change, and one element of climate change is obviously elevated CO2. So CO2 levels now are about, I don't know, about 450 ppm. Again, when I was doing my PhD some time ago, they're about 300 ppm. And although CO2 emissions, we know that over the next 10, 20, 30 years, in all likelihood, CO2 levels are going to increase. So we parameterized our model using data from two sets of plants. One set of plants grown under normal ambient conditions of CO2, then a second set of plants which we grew at elevated CO2 levels. This is because in Sheffield, we've got a whole suite of growth cabinets, which I'll show you perhaps in the second part of the talk this morning, where we can change or replicate virtually any parameter that you want in terms of CO2 level, light level, relative humidity, circadian rhythm. So these are growth cabinets that you can grow your plants, replicate essentially any climate that you want to around the world. And so in Sheffield, we can grow rice in cabinets under conditions which are sort of similar to what happens outside. Obviously, it's not totally replicating the lovely conditions in Malaysia, but we can also impose conditions which are not yet present in the environment. So we can grow the plants at, say, 800 ppm CO2, which is possibly what CO2 levels will be in 20, 30 years' time. Because if you're going to breed plants takes time, and if you wait for the climate to get to that level, it's already too late to make the plants to adjust to that environment. Within the model, what you do is you model the way that light reflects or refracts through tissue, because we want to measure or estimate what the light conditions are in every single modeled cell within our computational leaf. And similarly, they have CO2 that is diffusing into the system, and therefore we want to model what the local level of CO2 is around every single cell within our computational virtual leaf. And then finally, as I said, what we do is that within every single individual cell, there's what's called a metabolic module. So this is the basically simulating the process, the metabolic process of photosynthesis, and how well that photosynthesis, that metabolism works, depends on the local light conditions and the local CO2 conditions. So you can see this gets actually pretty complicated. And what the computer does is then integrate all that information together to give a final output, which is what you would measure if you went into the field and measured the rate of photosynthesis of a whole leaf. Okay, so the model contains a lot of information, cellular and subcellular level, and then it integrates outputs of every single cell to create a general output. So you can say this is the level of photosynthesis in our modeled leaf, and then you can compare the output of your computational leaf with what you measure in real life, and hopefully they come together, they're the same. So this is sort of model validation. And this is what's shown on the next slide. So again, don't worry if you, one, can't read these very well, and two, if you not, if these are new sorts of data. Essentially what we have, if I can find my pointer, the pointer has gone, pointer, oh, there it is, fantastic. Okay, so bottom left here, what you've got here on the y-axis, that is a measure of the rate of photosynthesis. And here, you've got the rate of photosynthesis depending on the amount of CO2 that you give to the leaf. Now, there are actually two lines here. You can hopefully see the red spots, and the red spots are what we actually measured in our rice leaves, in our chambers in Sheffield. So this is, if you like, reality. And then hopefully you can see there's some black lines here, and those are the outputs of the model that we created. And when I say we actually, I mean our colleagues in Shanghai created. And the bottom line here is that if you compare the pattern of the red dots and the pattern of the black lines, they're pretty close. They're not identical. There's not total superimposition, but nevertheless, the outputs of the model pretty closely capture the outputs that you measure in real life. Those are just two modeled data. What the computational model to create a whole field, if you like, a surface plot showing single spot. Again, I've lost my hip, but here you have a surface plot, and here's light, so that's high light to low light. Here's low CO2 to high CO2, and then here you've got the rate of photosynthesis, the rate of carbon assimilation. The computational model can allow you to predict what the output of photosynthesis is at every single light level and every single CO2 level in this field. Okay, so that gives you a prediction of how the leaf will function. It's every single curve, because measuring every single curve along here would take a long, long time. So that's the situation under ambient CO2, normal CO2 conditions. You do exactly the same thing for elevated CO2, which is shown here. Again, find the lost my measure again. And the right-hand side is the elevated CO2. And on the bottom right-hand side, what you can see is, again, measure of the assimilation rate against the different CO2 concentrations. The spots in red are the measured, and if you can see it, there's a black dotted line, and that's the output of the model. And again, hopefully what you can see here, the main thing is that the red dots and the black line, they're pretty much superimposed. Again, that indicates that the outputs of the model are capturing the real life outputs that you measure in a rice leaf. And then that's with varying the CO2 concentration, and this is varying the light intensity. And again, the red dots compared with the black line, they're not perfect, but it's pretty good. So what this tells us is that this model that we've created is capturing effectively the real life function of a leaf in our growth chamber in Sheffield. Once you've got those data, you can start to interrogate the model. And what you can do there is to say you have your model under ambient CO2, but you can then take the complicated equations and then substitute, for example, the level you measure under elevated CO2 for one parameter, put it into the equation for ambient CO2, but just change the parameter for elevated CO2. Again, it's reasonably complicated, but it allows you to sequentially go through every single parameter in your model, in your equation, and to judge, to estimate what is the contribution of that particular parameter to the change in photosynthesis that you see under elevated CO2. Again, this leads to a very complicated output. So again, don't worry to try and understand or read all of the detail. Essentially, what we've got is on the left-hand side here, the change of photosynthesis, the change of assimilation rate, and the different colors here represent individual parameters within the model. So for example, where you see light blue, that relates to bundle sheath properties. Where you see the purple, to mesophyll cell properties. The main thing to take home is, first of all, that some of these changes are actually positive on photosynthesis, some are negative. So what this tells you, actually, some of the changes that happens in a leaf, in a rice leaf, when you grow it under CO2, are actually, if you like, bad for photosynthesis. They're actually making photosynthesis work. You're getting a lower assimilation rate than we would want to see. It doesn't mean the plant is wrong, but that's the way that the plant itself is responding to that change of CO2 level. Also, what this graph tells you is that there's no one single trait, there's no one single magical element that you can change in a rice leaf that would make it perform better or worse under elevated CO2. The other thing to notice here is what you can't see, and again, since the slides are not ideal, this may be even more difficult to see, but there are some parameters. For example, here's something called leaf porosity, which is in green, which only occurs at a very, very small level in any of the things, any of the levels of CO2 change. And what this tells you, that this parameter, leaf porosity, is actually just not very, very important. So leaf porosity, that's a measure of the amount of airspace in a leaf compared to the amount of solid tissue. And so what our model is saying is that although you open up a rice leaf and it's sort of packed full of cells, it's very dense, the model says that actually you could probably remove a lot of that solid tissue and replace it with air, and the leaf would work just as well. Now, at first, that sounds a bit crazy, but actually, if you go in the literature and look at other research that is happening around the world, this is actually what a large amount of experimental data is also suggesting. So we have this idea that to improve photosynthesis, you need to pack leaves full of cells to photosynthesize. In actual fact, because of the way that plants work, you can actually often remove photosynthetic material from a leaf, but yet the actual total photosynthesis performance actually doesn't change that much. So these modeling data actually back up what other researchers have found or are finding outside. And then I'll just skip over that. The other point to take home is all the things on the left-hand side here relate to the structure of the leaf. Still, the main influence on photosynthesis is metabolism. So all the gray bars here indicate the the influence of changes of photosynthetic metabolism on photosynthesis under different CO2 conditions. And if you compare the scale bars here, that's at 6, that's at 1, then nevertheless, metabolism is the main driver for changing the way that plants photosynthesize under elevated CO2. Okay, and then finally, before I hand over to Jen, other thing you can start to do with these models is to start to create leaves that don't exist. Okay, so one aspect of rice leaves is they have mesophyll cells that are very highly lobed. So if you cut open a rice leaf and look at it under a microscope, the mesophyll cells are not sort of cuboid, they're not sort of elongated as you would say in a dicot leaf. They've got lots of lobes around them. And a number of researchers over a number of years have suggested this is really important. Again, this comes back to the fact that photosynthesis depends on diffusion of CO2. And if you have a very lobed cell, that increases the surface area to volume ratio, and therefore you'll think that would be good for something to diffuses into the cell. So what you can do with the computational model is to ask the question, is that true? Now, most rice leaf cells have maybe lobe numbers of maybe about 6 of area, 6, 8, 4. What we can do is to, in our computer models, create rice leaves that have lobe cells of 10 lobes per cell, or 12, or 16. So you can start to do things on the computer that, well, you could do experimentally, but it'd be very difficult and would take a long, long time. And so you're now creating leaves that don't exist and saying, okay, what happens? And the interesting thing for, again, this may not come out very well on the next slide, not very well. But again, this is a measure of simulation rate, and this is a measure of the lobe number, and this is changing in CO2 level. Now, we can't, there's actually is a color here, but essentially our data show that as long as you have mesophyll cells with a lobe number of 4 or greater, actually nothing much changes. And it's only when you get to mesophyll cells that have a lobe of 4 or less that for some reason, it's still not entirely clear why this happens, photosynthesis just crashes in the computational model. So again, exactly what's happening here, I'm not quite sure we totally fully understand, but just an example of once you've got a computational model, you can start to explore things and do things that experimentally would be incredibly difficult to do. And you can get some insight into whether something that the textbook says is really important is as important as people say. And so certainly here, our data indicate that as long as you have a certain number of lobes, more lobes is not worse, it's also not better. It doesn't play that much of a role. Which again, thinking about other projects that are happening in the rice world in terms of engineering rice, that might be quite informative. Okay, so basically to summarize the first part of the talk before I hand over to Jen, what we've done over the last few years is to create a model called eLeaf, which successfully captures photosynthetic performance under ambient and elevated CO2 levels. And by interrogating this model, you can start to get clues as to what is important or what's not important for a leaf in terms of its photosynthetic performance, both under ambient and future levels of elevated CO2. And the data indicate that leaf porosity actually plays a little, not much of a role. One of those traits actually you could increase, decrease, and the leaf would still function fine. And also that this idea that cell lobing is important or mesophyll cell lobing is important for rice, it's sort of true, but possibly not quite as in the way that people have written in the textbook, as in it seems you need a minimum number of lobes for the system to work. But once you've gone above a certain low number, say 4, the system works absolutely fine. Okay, so that's the first part of the talk. What I'm going to do now is hand over to Jen to tell you about a second part of the fundamental science that we've been doing, which links again into this idea of mesophyll cell size and shape. And again, some new insight into, what we think a rice leaf actually looks like. So Jen, do you want to come up and take over? Okay. Questions at the end. And where's my mic? Okay, someone put it on me.
Hi, thank you for inviting us. So I'm Jen, and as Andy says, I'm going to talk a little bit more about the rice mesophyll, which is the place where most of the photosynthesis occurs in the rice leaf. So this is when you're thinking about potentially improving rice yields, this is the part of the rice leaf that's most important, that is the bit that we are focusing on. So I don't know how much of a background everybody has in biology in leaf structure. So briefly, there's two main categories of plants, dicots and monocots. Dicots include, we've got here the little weed, Arabidopsis. It's the model plant. A lot of scientists work on Arabidopsis because it has a short lifespan. We know a lot about the genetics of it, but it's perhaps not entirely relevant to applied science. We've got crop plants like tomatoes, chilies, aubergines, okra. They're all dicots. Monocots are the grasses. So I've got pictures here of maize and of rice, also wheat. And the structure of the leaf, the inside of the leaf, is very different between these two. I would say at home, if you have a picture of a leaf in a picture that you would get, it's a dicot leaf. And you can see the mesophyll is split into two distinct sections. So you've got on the upper surface of the leaf, the palisade mesophyll with these long, thin vertical cells. And on the lower side of the leaf, the spongy mesophyll with smaller cells, they're more irregularly shaped, and there's a lot more air space in between them. Dicot leaves typically sit horizontally. So it makes sense that there's a difference between the top and the bottom. The stomata, those pores that let the CO2 in and the water out of the leaf, they mostly are on the lower surface of the leaf to conserve water. So we've got here specialized shapes within the mesophyll. You've got these specialized palisade, long, thin cells. They help to direct the light down into the rest of the leaf, into the lower section of the leaf. This part of the leaf is probably best designed for photosynthesis, for gas exchange. There's a lot of space in there for the gas to reach the cells, and the chloroplast, which is where the photosynthesis is happening. This is the cross-section through a rice leaf. The top one was tomato, by the way. So this is a rice leaf, and the mesophyll is uniform. The cells are all the same. At least that's what the textbooks say. So I've looked at a lot of sections of rice leaf across all of this work that Andy was describing while we were creating the parameters for the e-leaf model. And the more of these rice leaf sections I looked at, the more I said, they're not all the same. Those cells are quite different. And I think I can see patterns there. I think I can see something that is the same across every leaf I've looked at. So this was actually a fortuitous thing that came, a lucky thing that came out of the pandemic, is Andy, we're in lockdown, nobody's allowed to leave our houses. We were in lockdown, but we still had students. And Andy asked me, could I think of a master's project that didn't involve going into the lab, that could be done entirely from home? And that was where this thought that had been in the back of my mind, I said, I think I've seen this pattern. I said, I know what we can do on our computer with a master's student who is willing and has lots of time on their hands. And this is where Yang, who is a Malaysian master's student, stepped in, and I gave him so many images of rice leaves that we already had, that I was able to send to him, and between us we worked out exactly how to look at these cells, how to measure them, and the number of cells that Yang drew around very patiently over the course of his master's project is phenomenal. So to see if this truly was a pattern that was represented across the varieties, the different varieties of rice, we looked at 6 different rice varieties. The 3 across the top here are all Malaysian varieties that were kind of given to us by Adam. We've got Malaysia, which is quite an old variety, MR220, and the fragrant rice MRQ76. And we also had seeds for some wild varieties of rice. So these are not cultivated. We've got Oryza latifolia, Oryza punctata, and Oryza meridionalis. And you can see that the growth structure of these plants is quite different. Particularly latifolia and punctata are really, really large with quite wide leaves. The Oryza sativas all look broadly similar. Meridionalis looks really grass-like. When you look at them in cross-section, there are similarities, there are differences. You can see the differences in size, perhaps, but you can't tell a lot just by looking at pictures. So this is where measuring these things and drawing around the cells comes into play. These are the layers that I thought I'd seen. So there's a layer in the middle here, which goes directly between the 2 minor veins, that goes in a roughly straight line between them and isn't disrupted. You've got a layer along the top, which touches the epidermal cells, the top surface of the leaf, and similarly a layer along the bottom here. And then we've filled in the gaps. So any cells in between these 2 layers, we've called layer 2, and any cells that occur between layer 3 and 5 are layer 4. All the rice leaves that we looked at had at least 4 layers, so layer 1, 2, 3, and 5. Layer 4 wasn't always present, but usually was. So using a program called ImageJ, we were able to draw around these cells, and then it gives you parameters of the shape that you've drawn. One of the parameters it tells you is the size of the cells. Each of these graphs represents a different variety. We've got the 3 Oryza sativa varieties along the top, and then the wild varieties here along the bottom. And each graph is split up into those 5 layers. Layer 1, 2, 3, which is that middle layer, layer 4, and layer 5 at the bottom of the leaf. And I think it's fairly clear from this data that there is a difference in cell size between these layers. And we found that the middle layer there is always the largest, on average, always has the largest cell size. So this starts to show that perhaps I was right. The mesophyll of monocots, or rice in particular, is perhaps not uniform. We're seeing a consistent pattern across 6 different varieties. So then I wanted to look at shape. Is there a difference in shape of these cells across the different layers? And to measure shape, we looked at circularity. So how similar is this shape to a circle? A circle has a perfect circle has a circularity of 1, and anything that is less circular, I've demonstrated with some just cartoon rice cells along the bottom, has decreasing circularity. So it both represents how circular or oval the shape is, but also how wiggly the outline is. And again, we found that those middle cells stand out. They are the least circular, so they are more oval, and they're also perhaps more lobed. And again, although it's only significant in Oryza latifolia, that pattern that shows that the layer 3 cells are always the least circular is consistent across the 6 varieties. And the last thing we thought about in the dicot leaf that I showed you, the palisade cells are vertical, and the spongy mesophyll cells are more randomly orientated. So I wanted to look at the angle of the cell as well and see if there was anything that was a pattern in the rice leaf cells. Again, using this image J, we were able to automatically tells you the longest axis through the shape and what angle that has. We then, this is the measure of the angle. We adjusted those angles so that they were all within a range of between 0 and 90 degrees. So it's just telling you how far off vertical it is. That everything is leaning in the same direction now, just so it's easier to compare. 0, the degree of 0 is entirely horizontal, and 90 degrees is a vertical cell. And this is the graph of what this small selection of cells would look like. So we've got 1 that's horizontal, 2 that are nearly vertical. So we just broadly categorize them like this. And there's a lot of data here, but you don't need to look at the data, you just look at the patterns. So what we've got is the 6 varieties along the top and the layers of the cell. So layer 1 at the top of the leaf, 2, layer 3 in the middle, layer 4, and layer 5 along the bottom of the leaf. And this shows the angle of the longest part of that cell. And I think it's quite striking that those middle cells are far more horizontal than most of the cells. There's perhaps a slight tendency for these top layer of cells to be more vertical. And these cells along the bottom, you get an average of 45-degree angle, which really reflects the fact that they're quite square in shape. So the longest angle through a square is that diagonal. Another way of looking at the same thing is to look at the average shape of each of those cells, taken each cell from and superimposed it on top of it, each of the cells superimposed on top of each other. So it's the same as before. This is the layer 1 cells on the top, the middle layer here, and the layer 5 cells on the bottom. And I think particularly if you look at Oryza latifolia, you can see that there's a massive difference in cell shape and size depending on where they are in the leaf. Also this one, Oryza latifolia and punctata, these two particularly have the most variation within the leaf. There's a real variety of cell shapes and sizes in those leaves. So to sum up, and we're really just trying to better understand the mesophyll. So I hope I've convinced you that we've seen this pattern, that the layer 3 cells along the middle are larger, less circular, and more horizontal than other cell layers. So why do we care? I feel that if you see a pattern that's replicated so often in all these, across this broad range of rice varieties, perhaps it's important. Perhaps this pattern has evolved for a reason. Is there something about this pattern that is helping the rice leaf? Could those layer 3 cells, those larger, more horizontal cells, could they be specialized for photosynthesis? Or is it, we don't know the answers. These are just questions. Are larger cells better for photosynthesis? They have a larger vacuole. Is that important? Are layer 1 and layer 5 cells, those on the outside of the leaf, are they acting a little bit like the palisade cells in your dicot leaf, directing the light into the center of the leaf? Possibly. Is it something to do with the amount of airspace, as Andy was talking about earlier, the amount of airspace that can access those cells so that photosynthesis can happen? Is there something that we could look at tweaking, making rice leaves more productive based on these patterns that we've seen? I'm going to skip these, I know we're low on time. We're also wondering if cultivation has had a role to play in this, because our wild varieties have a lot more variation than the cultivated varieties? Is there a story there? Is there something we can learn from the wild varieties and bring back into our cultivated crop plants? I want to briefly show you, I know, as I say, we are a bit short on time, but I want to show you something else that we're looking at to try and hopefully in future make our models even more accurate, and also just improve our understanding of the mesophyll, that key part of the leaf where photosynthesis happens. We want to look at whether there are other gradients in cell size, perhaps going in the other direction, so not looking at the top to the bottom of the leaf, but looking between the mid veins, the minor veins. Is there a difference in cell size in that direction? Is that important? But to really understand the structure of these cells, we want and have wanted for a long time now to be able to image them in 3D rather than always looking at two-dimensional sections. Another postdoc in our group, Matt Wilson, has recently in wheat managed to get some 3D structures of mesophyll cells, and it was really informative to look at the cells in 3D. This is taken from his recent paper, and he helped me. And can I make this play? Yes, this is a rice confocal image, and Matt helped me use his technique that he'd used in wheat to extract the structure of some rice mesophyll cells. The cells in rice are a lot smaller than in wheat, so this is fiddly, quite tricky, but also really quite impressive and exciting that we have this potential to get the 3D structure of mesophyll cells in rice. So this is something that we're going to be spending quite a bit of time on the next chunk of time. This is what we're going to be focusing on to really understand the size and shape of these cells in a three-dimensional way. We're also at the same time going to look at the bundle sheath cells. So this is the mesophyll that I've been talking about. Another important set of cells, which Andy did mention in the modeling section of the talk, the bundle sheath cells. So these surround the vasculature. They are photosynthetic cells. They have chloroplasts, but they're also involved in transporting the products of photosynthesis into the vasculature, into the veins, and then throughout the rest of the plant. Oh, why does that one not play? So this is another confocal stack, and this time I've highlighted the bundle sheath cells. When you look at them in a two-dimensional section, they look like circles, as I just showed you. But when we and they are there space between the features of the cell. So this is a heat map here, which is telling us the surface area of the cells. The lower surface area are a dark blue. Let's skip back a bit, that's better. So the cells with a lower surface area are a darker color, and the lighter colored cells here have a larger surface area. So this is you can also extract cell volume, so the size, the three-dimensional size of the cells. And again, we've got the smaller cells going up to larger ones here with the lighter color. And this is that same data again, the cell surface area. These are IR64 rice leaves. The earlier I showed you some data from latifolia, which was big, which had the biggest cells. It's a nice one to look at. It's a nice one to compare with IR64 because you've got a difference there. They have bigger cells. The leaves are bigger, but the cells are bigger too. So we wanted to have a quick look at latifolia. I want to point out that this is very, very new data, and it's perhaps not perfect. I did this the Friday and we flew on Saturday. So it's new. And it's also, I'm still learning how to use this program. But this is, One of them played. Where is it? So yeah, this is Oryza latifolia. And again, the fact that we've managed to get this confocal image all the way through the thickness of the rice leaf is very new for us. We had to use a newer confocal microscope to get these images all the way through the thickness of the rice leaf. We've only really been able to do that for the last, I think in the last 3 or 4 months. So this is latifolia, and it's kind of hard to look at it on its own and see whether it's the same or different to IL-64 that we were looking at before. So this is the cell size, the cell volume of IL-64, and then compared at the same scale to latifolia here. And when we look at the scale bar here, it goes from red for very large cells down to blue. And I think the difference is quite striking. And no one's surprised that latifolia has bigger cells than IL-64 because it's bigger, it has bigger cells. But the important thing here is that we can measure that difference and we can quantify that difference, and that's new. We can do this in 3 dimensions. And you can take these heat map pictures and extract numbers from them, and then plot that on a graph, which maybe helps to visualize the difference between 2 things. So you've got a range of cell sizes here in latifolia. And IL-64, as we saw in the mesophyll, latifolia has a larger range of cell sizes, more variation. So it's true in the bundle sheath as well as the mesophyll. And I've got that the same stacks, the same images there, but this time looking at surface area. And it's a very similar pattern when you look at the numbers. Interestingly, when you compare the surface area to volume ratio, so now we're looking at the shape of the cells, they're actually not that dissimilar between latifolia and IL-64. So the cells have changed size, but not shape. And that's another interesting thing to think about. Why might that be? How can we use that information to better understand photosynthesis? So in summary, we have the model that Andy talked through, we've created this model based on true leaf structures, and it successfully captures photosynthetic performance under ambient and elevated CO2. So we can recreate real life photosynthesis on the computer. We've interrogated that model, and one of the conclusions we've reached is that leaf porosity is not as important in photosynthesis as you might think, and that's really key. There isn't a massive importance on the degree of cell lobing, and again, that's surprising. It doesn't perhaps match what people might have expected. We've seen this pattern in the mesophyll. We now need to think about why that pattern might be there and try to understand the function of the pattern. And as I say, we're really quite excited about the 3D analysis of the rice mesophyll and the bundle sheath. This is going to allow us to really understand that structure and then better parameterize our models. Which leaves me to say thank you to everybody. And it's a big group of people that have done all of this work. So in Sheffield, there's Andy and myself. There's a large group of people working on the initial parameterization of the model, doing all of those measurements. Matt Wilson has helped with the imaging, and Yang is the master's student who I mentioned, who drew around so many mesophyll cells. In China, we have Yi and Jing Wang, who have done all of the computational work, and they put up with our questions when we don't understand what the computer model is trying to tell us. The people in Nottingham, I lost the cursor. There we go. The people in Nottingham helped with did the CT imaging, so that large scale imaging that we did to look at the airspace within the leaf. And of course, everybody at UPM who now. and should thank the people who've given us money to do this work. That's it. Thank you.
Oh, I think we're ready to take questions. If there's questions.
Yeah, so I think we can take questions now. So this is the first one, right?
The first talk, yeah. So there's the questions for the science bit, then a break, and then the translational stuff, and then more questions. Is there a question?
Kalau nak tanya bahasa Melayu boleh. Saya boleh marah. Boleh kita. Asyik kita berkenal sama lain.
Of course, yeah, sure.
I've got a question actually. So it seems like the biochemical is more important in determining the photosynthesis rather than the structure of it. Is this true for the dipole?
We don't know that, but I would expect that is the case. So again, if that's not so surprising, I mean, there's been a number of major research projects, very successful research projects, mainly out in Illinois, where they've been re-engineering photosynthesis to, for example, decrease photorespiration or increase various aspects of the carbon density cycle. I say they've been very successful. So clearly, metabolism is important. First, the question that we were addressing in this project is, to what extent is leaf structure important or not at all? And I think our results would suggest it is important, and that's about 10% or something. So it depends, yeah, how do you define important? It has a role, that's very clear, but perhaps surprisingly is that just how flexible a leaf can be, and that our data would suggest there are some elements of leaf structure that can really change a lot, and the leaf just accommodates to that, and it still functions perfectly well. So yeah, how surprising that is, that's a matter for the community to discuss. But I expect in dicots it would be the same. My gut feeling, by the heavy data on that.
Yeah, because many of the important crops are actually dicots. Like the aubergine, okra, these other if that somehow is true for them as well.
I would expect that metabolism at the end of the day is always going to be absolutely key, clearly, because it is at the end it's a metabolic process of CO2 fixation.
Right. Okay, thank you for that.
Thanks for the great help. So basically, I noticed the model mainly focusing on the aerial parameter like the irradiance and the carbon dioxide concentration. How about water conductance, the water ability?
Thank you, Joe. we avoid water conductors. That's a very fair question. So you've got obviously the vascular conduct, how water gets to the actual cells is really important, and there's lots of information on there. And also in the model itself, no one's asked the question, stomata are not explicitly modeled within the leaf. We sort of make the assumption that water sort of leaves the leaf and comes in the leaf via stomata. So it is a first go, a first approximation, and it captures, we think, that the basics of how a leaf actually works. We are absolutely right. If we could also then introduce another element of how water flows through the vascular system and out of the vascular system, and then that will also influence to some extent how the system works. But that's not in the model. You're absolutely right. I guess our only defense there, you've got to start somewhere, and trying to keep it simple and making it work, and then adding on more complicated stuff. That's a fair question.
All right, thank you for that. Thanks, Andy.
Andy, can you give your opinion about the current project, about the C4 project?
Thank you, Adam. I mean, so I'm not in any way directly involved in the C4 project. So perhaps again, perhaps for the general audience, So rice has something called C3 photosynthesis, which is like the standard photosynthesis that most plants that you see outside use. However, there are a number of plants which have a boosted form of photosynthesis called C4, where they initially capture CO2 not using the enzyme rubisco, but by another enzyme called carboxylase, which is much more efficient than rubisco in capturing CO2. And then they transport that CO2 from the mesoscale to the bundle of sheath cells, which again talked about, where the rubisco is located. And what C4 photosynthesis does is to increase the local concentration of CO2 in the bundle of sheath cells, and that makes the whole process of photosynthesis much more efficient. And so I don't know when the C4 project started, it must be 10, 15 years ago. The big idea is that if you could make rice, a C3 plant, into a C4 plant, then automatically you'll increase the yield of rice by 25, 30% at no extra cost of fertilizer or anything like that. So say that's, I know the number of people involved in that project. For example, Paul Frick from Sheffield was involved for a long time. And it's now been led by Jane Langdale at Rochester. So the number of papers have been published, so they're making progress towards it. But as yet, as far as I'm aware, they have not yet been able to create a C4 rice. They've been able to gain a greater understanding of what you need to make a C4 rice. So all of the genes that are needed, where those genes need to be expressed, an understanding also of leaf structure, and that's possibly where our work can make a contribution, because for a C4 rice to work, you have to have a certain number of cells between the vascular tissue. So you have vascular tissue, then you have bundle sheath cells, and you can only have a maximum of maybe 2 or maybe 3 mesoscale cells in between. Whereas in rice, normally between the vascular bundles, you have maybe 10, 12 mesoscale cells in between. So if you wanted to engineer C4 rice, you've got to decrease the number of mesoscale cells between the vascular bundles. And what our data indicate that actually that shouldn't be a bad thing to do, because, for example, you can imagine making bigger cells which have more loads, and our data suggests, yeah, that's fine, it's no problem. You can change the number of mesoscale cells in a rice leaf, and that shouldn't have a major effect. So I suppose our data indicate that theoretically, again, they provide theoretical supports that for what they're trying to achieve in the C4 project in terms of rice leaf anatomy, it shouldn't be necessarily detrimental to the leaf. But whether you can get or breed or select or engineer a rice leaf that has that particular architecture, that's another problem that we're not addressing, which I know is a focus for the folks who are working on the C4 project. So I'd say I'm not directly involved in that project. They're making progress, but as far as I'm aware, they still haven't really got to having a prototype C4 rice in their hand. And I think actually the leaf architecture is in some ways the more challenging part of that project. The metabolism bit, they know what to do there, and I think with the modern breeding methods and the methods of using gene editing, that's doable. Changing the structure of a leaf actually is sometimes actually the more challenging bit. I think that's where they're trying to make more progress. Okay, that's as much as I know about the point.
Thank you, Ali. So do you think that the current work that you are having now can somehow be and contribute to C4?
I think it provides a theoretical basis that for what they're trying to do in terms of rice leaf architecture, it's not a bad thing. It's not a case. If we'd shown that, for example, if you make a rice mesoscale cell, I know, double the size, or you make it increase the load and the whole thing crashes, that certainly would, our data suggest that's not the case. And there's a lot of, and the data from the porosity indicate that, again, changing the amount of airspace or the amount of tissue actually in the rice leaf, you can play around that quite a lot. And the modeling suggests that, yeah, the rice leaf will soak it up and it will accommodate. So again, it is a hydroplasticity within the system, so that engineering it should be feasible. Whether it's practically easy to do is another matter. At least theoretically, it should be fine.
Thank you. That's very informative. So, yeah.
Okay, hi, good morning, Professor. There's a question for the Zoom audience. Okay, here is the questions. Are the mesophyll cells structure or function have any relation or influence in drought, salinity, submergence tolerance in rice?
I take that one or you?
Okay, go for it.
I'll go for it. Okay. So the question is the role of the mesophyll in drought tolerance. Is that the basic?
And does the structure has any relation?
I mean, certainly work that we've done in Sheffield in Arabidopsis and in wheat suggests that there is a relationship. There's a relationship, but we don't really understand the function. So that in take the wheat data. So wheat, modern wheat that you red wheat is hexaploid, and that's derived from breeding from tetraploid and from diploid wheat. And from a series of experiments that actually Matt Wilson, another postdoc has done, if you look at the stomatal conductors, so effectively the amount of water that's being lost by the plant, there's a relationship with the ploidy level of the wheat, and that also correlates really nicely with the porosity of the wheat. So there is a relationship between the internal mesophyll structure and the water use efficiency, the ability of the plant to control water loss. And that also seems to be linked to the stomata structure and density on the leaf. So how it actually works, we would really like to know. We have sort of hand-wavy explanation. So there is a relationship. I think that's clear in wheat, and also we've got nice data in Arabidopsis that it's true. But how it works, we don't really know. Whether it's related to the internal exposed surface area of the mesophyll, that's really important, because that's the area, that's the surface area from which water escapes from cells into the internal airspace that's out by the stomata. So we think that by altering the architecture of the mesophyll, what you're really doing is changing the surface area to volume ratio of your mesophyll. And again, this is all to do with gas diffusion. And with any gas diffusion system, if you're changing the surface area to volume ratio, then automatically you will influence the rate at which gas is lost from the surface. So that's about as far as we've got. So the answer is there is a relationship. What the functionality is, exactly how it works, we don't know, but it's certainly something we are investigating and want to follow up on. But we're doing that mainly in wheat, I must say, rather than in rice.
Is that your question? Okay. So I think that's all for now. So we're going to have a short break for 15 minutes. So you can have some more buns from the outside if you haven't. So we'll come back and continue for the second talk around 10:40, I think. Right? Okay. So I'll see you in a bit. listen to it attentively.
Thank you.
Oh, it's with me.
Okay, thank you very much. So welcome back everybody to the second half of the show. This half will be shorter than the first half, and will be more a discussion about some of the progress that we've made, more on the translation of the fundamental science that we've been doing as part of the project that we talked about in the first part of the morning. And then finally, if there's time, just at the end, I'm just going to say a few words about international interactions and collaborations, and certainly my experience, and yes, what works, what doesn't work, and the challenges that can be involved when you start working with people from around the globe. Okay, so Jen, just to remind you, the project started as funding from this GCRF, this Global Challenge Research Fund from the UK government. And the project that we discussed in the first part was, although it has eventually an applied aspect, is actually it involves pretty fundamental science, so understanding how a leaf works. But what the funders really want to achieve in the end is something that will help people in the real world. The modeling approach that we've discussed, that will take time. Okay, it's a long-term project. As soon as you get into identifying traits for breeding, that's not a quick win at all. So as part of the project, we had to grow plants under elevated CO2. Now, how do we do this? And again, this is not a great diagram, but this is a picture of our plant growth facility at the University of Sheffield. So basically, it's underneath a car park, so it sounds a bit strange, but we have two levels of growth cabinets there. And these are these sort of blue boxes here, and I can't remember how many we've got now. It's like 40 or 50 in various sort of sizes and shapes. And these are fantastic facility to have, but it doesn't come cheap. So this is probably like, I know, 12, 13 million pounds worth of investment to create these facilities. And what they now allow you to do, is to grow your plants. And again, you can't see this very well, but there's various LED lights here, so you can change the light conditions, the light quality, the light intensity. We've also got sort of robots. Again, this is, you can see this here, this little gantry here. This is basically a motor that goes back and forward, and it can view your plants, measure whatever wavelength of light you want to do it all automatically, and screen lots of plants. And these facilities, so this is the equivalent of a rice paddy in Sheffield, which doesn't look much like a rice paddy in Malaysia. But we've got lots of rice plants here in a chamber, growing pretty well, setting seed. And what this facility then allows you to do is to do some quite sort of fancy experiments that you really would find very, very difficult to do outside in a typical rice field. So one thing that we did is almost a side project, or an observation that we made during the main project, was as follows. So to parameterize the model, the e-leaf model that I talked about, we had to grow the plants under either elevated CO2 or under ambient CO2 to look at the structure and the metabolism of the rice plants under these different CO2 conditions. And then we used those data to inform the model. And we then asked the question, okay, we started to see some things happening in the mature plants, in particular related to tillering. So tillering essentially is branching in rice. So it gives it controls the bushness of the rice plant that you get. And what we started to do was to do experiments where, instead of keeping your rice plants all of the time under elevated CO2, we asked the question, okay, what happens if you grow your rice plants under elevated CO2 for a short time and then transfer them to ambient normal CO2 conditions for a longer time? And hopefully this picture comes out. It's not too bad. So on the right-hand side here, you've got a rice plant that's been grown under elevated, so 1,000 ppm CO2 throughout its whole lifetime. And then on the left-hand side, you've got a plant that's been grown under ambient, so normal CO2 conditions for the whole time. And hopefully what you can already see by eye is that this rice plant here on the right-hand side just looks a bit bushier, got more sort of stalks, more leaves than the plant on the left-hand side. And in between, we've got a series of plants where we grew the plants under elevated CO2 for 21 days, and then moved them of elevated CO2 and then moved them to ambient, or 28 days under elevated CO2, moved to ambient, or 35 days and moved them to ambient. It's probably not so visible by eye, but if you plot the number of tillers per plant against the treatment that you had, then what you can find, the stats aren't on these, but they are statistically significant. Under elevated CO2, you get a higher number of tillers per plant than if you grow them just under ambient CO2. If you grow them under 21 days elevated CO2 and then transfer to ambient, then in the mature plant, you get about the same number of tillers. But as soon as you start growing them for like 28 days under elevated CO2 and then transfer to ambient, you start getting mature plants that have a much higher number of tillers than if you just grew them under ambient. So again, what you're doing here, you're growing your plants for a relatively short period of time, 3 or 4 weeks under a particular elevated level of CO2, then moving them to an ambient CO2 chamber and letting them grow to maturity, then counting the tillers. So these experiments are first of all done with IR64, which is our sort of standard variety. And again, it's a bit blurry, but hopefully what you can see, and this is certainly shown here on the lower slide, it's clearer, that in IR64, you get a highly significant increased number of tillers if you grow the plants for a short time under elevated CO2 and transfer them to ambient. And this seems to work with virtually all the varieties we've looked at. This is, again, Malaysian varieties provided by Armin, so it's MR263, MR219. And yeah, perhaps the differences are not as massive as you see with IR64, but nevertheless, exposing the rice plants when they're young to elevated CO2 and then moving them to ambient CO2 leads to a long-term increase in tillering in a range of rice species. Now, why would anyone be interested in the number of tillers that a rice plant has? Again, I know you know more about this than I do, a number of you, but basically the number of inflorescences that you have on a rice plant is going to be limited, dictated by the number of tillers that you have. And the number of inflorescences that you have will limit the number of panicles that you can possibly get from a rice plant. And the number of panicles that you can get on a rice plant will set the boundary, will set the limit for the maximum number of seed number that you can get. Now, it doesn't mean that just because you have more tillers, you will get more seeds. Biology is not as simple as that. But if you think about the system as a whole, the maximum number of seeds that you could potentially get from a rice plant will be dictated by the number of tillers that plant forms during its lifetime. So this is one of those things that it wasn't sort of a main aim of the project, but again, this is how science works. You run an experiment, and then the people running the experiment see something, think, oh, something's happening there. You then follow it up, and yeah, it turns out to be true. And then you think about this, okay, could this be useful? We know there's a relationship between tiller number and potential yield. So we've done this all in these lovely, fancy growth cabinets in Sheffield. Could this observation at all be translated, applied to the field? Okay, so the basic part of this translational part of the project was, can you increase the tiller number in the field, in real rice plants, by exposing them to CO2, high CO2 for a short time? And if you get more tillers, what is the actual outcome on yield? So as I said, in Sheffield, we're very fortunate. We have this fantastic growth facility. And I say there's actually not many places in the UK have this facility. And if you think about actually growing rice in its true habitat out in the paddy, this is not how you're going to get elevated CO2. So, and this is really now referred to work that Armin and colleagues have been doing for the last couple of years, is to how would you test whether this is going to work in the field? And so this is a mock-up, a cartoon of a self-made growth cabinet that Armin creates in Malaysia. And in some places, this is actually quite straightforward because basically a growth cabinet has a light source, and now LEDs are really cheap. You can get a whole array of LEDs, different wavelengths. So setting up a light cabinet is relatively straightforward. In Sheffield, obviously, we have to normally raise the temperature to 28, 30 degrees and then control the temperature because that's not the normal temperature in Sheffield. Again, in Malaysia, it's pretty warm most of the time. And so with a few fans and stuff, you can set the temperature of a cabinet moderately well. In Sheffield, we increase the level of CO2 because we buy CO2. So a big van comes around with canisters and delivers the CO2. It's all very expensive, but it's all very, very tightly controlled. So the question really was, how would you expose your young rice seedlings in UPM to elevated CO2? And we, again, Armin and colleagues, tried various things. And the way that actually works quite neatly is to use what are called mycelium bags. So basically, mycelium bag, you have a bag of substrate and you prime or dose that substrate with fungal mycelia. And mycelia, as they grow, they respire. And again, because you're in a lovely warm climate, the CO2 levels produced by the respiration of fungi, the mycelium, is actually pretty impressive. So again, this is just a picture of one of these mycelium bags here on the right-hand side. Lots of fungal hyphae here respiring, and this becomes more important later. Those fungi at the end produce fruiting bodies, and some fruiting bodies are really very tasty. These are oyster mushrooms. Again, the reason for choosing oyster mushrooms becomes clear soon. So first of all, does it work? And what we've got here is a trace of CO2 levels in red. And yes, it's a living system, so it changes between light and dark. So this is, again, different from what we do in Sheffield because, again, we've got CO2 canisters and supply pipes. We can keep CO2 level pretty constant all the time at whatever level we want. So it's not the same as what we do in Sheffield, but nevertheless, it's very clear. You get actually a pretty astonishingly high level of CO2 being generated by fungi. It's really quite shocking or impressive. So basically what you can do here is to have a chamber with these mycelium bags generating CO2. That CO2 then lifted by a fan into the growth chamber part where the little rice seedlings are growing happily under the LEDs. And you essentially try to replicate the experiment that we did with our nice growth facility in Sheffield. And does it work? The answer is yes, it does. Again, the differences are not as massive as we see in Sheffield, and it does probably vary depending on the cultivar. But nevertheless, if you look at the number of tillers and the young seedlings after being exposed to CO2 for like 3 weeks or so, then with the elevated CO2, you do get an increased numbers of tillers relative to what you find with rice plants grown in the same conditions, just without the mycelium bags being produced. This is quite encouraging. And so the next thing is, okay, we can now generate rice plants with more tillers. We want to put those out in the field, grow them up, and see what happens to the yield of those plants. At this point, you have a global pandemic. And I must say that I'm sure everyone in this room involved in research, their research has been really badly affected by what's happened over the last 2 or 3 years. And again, this is an observation from what's happening in the UK. I somehow have become a more senior academic. We're very, very aware of the challenges there's been for a number of younger researchers who have, set up an experiment. There's been lockdown, you lose the experiment, you come back, set the experiment up again, it's another lockdown. It's been really, really tough and really frustrating for a whole load of researchers. So I won't go into the details of the whole process of the last year and a half. We've been struggling to try and get a complete experiment done. Suffice it to say that part of the reason why Jen and I are here this week is that we were just up in Mardi yesterday, looking at actual plants in the field that have been treated with the mycelium CO2 that have higher tiller numbers. And these are data from the field from Adam. And again, it's a MR219. And the data so far look quite encouraging in that, again, on the elevated CO2 is on the right-hand side, that the plants in the field have a higher number of tillers than the plants that have been kept just under ambient CO2. And we're at the stage now where the plants are essentially mature. I think the harvest is happening sort of as we speak now. And so hopefully in a week or 2 weeks, we'll actually eventually have field data as to whether this what we call CO2 priming, it certainly leads to an increase of tillers. Does that increase of tillers translate actually into an increase of yield of those plants? So in some ways, it's been frustrating to take so long to get to that stage, but we now hopefully, the pandemic's not over. That's absolutely clear, both in the UK and I'm sure here in Malaysia. We still have COVID, but we're at a stage where it's hopefully under control to some extent, and we can actually start doing completing experiments, especially long-term experiments, where the pandemic has had a real major negative effect. However, during the pandemic, obviously we couldn't get into labs, couldn't do stuff, but it doesn't mean you just sit at home and twiddle your thumbs. You can still do some analysis. And so this is sort of an idea of what could happen, not saying it will happen. And just, again, it's coming down to what the fund, the Royal Society were interested in is are there novel ways of helping rice farmers, essentially? And so what we've done, again, is really Armin and colleagues in Malaysia, just trying to work out, is this a feasible approach? Okay, we know that in the biology, it's hoped it's going to work, that yes, you can get little rice seedlings with more tillers. And if the data pan out as we're sort of hoping, we might be able to get more yield, but is that actually economically viable? Does it work? So if you think about it, the nice thing about here is that it's the whole thing. The key thing here is the mycelium, the fungus, and that uses lignocellulosic waste. And I think in Malaysia that's a pretty cheap resource, right? There's a lot of forestry going on, producing waste, which can be used as a substrate. That produces CO2, and then that CO2, hopefully, again, we're still waiting for the final data, leads to increased tillering, which then translates into increased rice yield. And as far as I understand it, in the system in Malaysia, there's a pretty standard rate of rice, of payment for rice. So that will translate into increased income for the farmer. Obviously, the key thing here really is how you expose those young rice seedlings to that elevated CO2. And I think we're still working on that as to how that could be scaled up to a process that would be amenable to farmers or communities of farmers using it in the villages. The nice thing here is that as well as the CO2, the mycelium itself is producing a product of value, mushrooms, which the farmer can sell and get income from. And then also, once you've grown the mycelium, what the fungus does as fungus is to break down lignocellulose into essentially compost. And this is actually real life bags of compost generated from the mycelium growth. And again, that has value for the farmer, either to be used in the fields or to be sold as a product. So again, our initial analysis is that actually there's a certain investment required. That's absolutely clear to get the process up and running. But once that investment had been made, then assuming that the increase of rice yield is what we expect it to be, then actually there's an economic return after a few years in terms of the combination of income from rice, income from the fruiting bodies that you are using to make the CO2, and the income from the compost that you're getting or deriving from that breaking down the CO2 as well. So those experiments are still ongoing. We'll hopefully be able to report on the actual values that we get from the rice very, very shortly. Now, again, this is a description of how research works and how sometimes you can perform an experiment and you observe something which is unexpected and you don't quite understand what's happening, but you can still use, if you like, bad results to actually make progress. So in the next part of this talk, I just want to talk about briefly about something else that with our project has sort of sidelined into in an unexpected fashion. And this came about because on the left-hand side here, I'm sure this is the way that most rice farmers would grow their initial rice seedlings before planting out in the paddy. So really high density in trays. Now, for the CO2 treatments, what we did without really thinking about it, I must admit, was to grow the rice seedlings at a much lower density. And that was just simply because of the way that we were growing and treating the rice in these artificial chambers. And it led us into experiments or ideas about whether there is an optimal density for initial sowing of rice seeds in rice agriculture. So again, this is a picture of some, again, not a great picture. Again, this is linked to the pandemic. We're trying desperately to get some experiments through when things were in lockdown. And for the priming experiments, these are done in pots, in glass, in shelters. We'll grow the plants at low density and then expose them to high CO2, so that's the elevated CO2 or ambient CO2, and then grow the plants in pots and then measure the yield and measure how well the plants were doing. At first sight, these data were very sort of disappointing in that on the left-hand side here, you probably can't read it very well, but this is number of tillers per plant, and this is the yield per plant. And there's no difference between the elevated CO2 and the ambient CO2. However, if you look at the absolute numbers of tillers on these plants, it is, compared to normal, just mad. They're just making so many more tillers than a normal rice plant ever would. And similarly, if you calculate the yield from these rice plants, and again, it's way, way higher than would be normal. And that is summarized in the next slide. So again, I'm sorry about the quality of the slide, but on the left-hand side here, this is again, I'm sure lots of people were doing this during lockdown. You're doing sort of meta-analysis, going through the data that's out there in literature. So here's data taken from a whole load of literature from different parts of the world where people have done sort of comparable experiments. We grow rice in pots under shelters or glass houses. And then on the right-hand side here, these are the data that we obtained here at UPM using this priming system where there wasn't actually much difference between the ambient CO2 treated and the elevated CO2 treatment, but the absolute levels of yield were clearly markedly higher than what other people in other parts of the world, including Malaysia, have been finding, where they've been growing the plants under like normal ways. And obviously, when you do these experiments, you think, okay, what is the difference? What is the variable between these plants here and these plants here? And the most simple thing to think about is the density at which we were putting the plants during that very early stage of germination to get them to a stage before you transplant them from the nursery into the paddy. So we're still following up on this, and it's led to actually basically a field trial that's still ongoing, and Mashita has been very heavily involved in this. We'll go to rice farmers, this is in Sabah, who still grow rice in a very sort of traditional way, so a lot of manual input, and getting directly in contact with rice farmers to see if they can set up their own experiments. So, and what happens if they start this early process of rice growth, this early phase where you have them in the nursery, but instead of packing the seeds in at really high density, you do it at a much lower density. And our idea is here, what you're doing is you're giving that an early vigor to the plant, so nurturing those seedlings in very early stage. How does that affect how the plant grows later on? So again, for a Malaysian audience, you'll know where Sabah is. This is more for a European audience, in case they don't know. So this is where the trial is being held. And the other nice thing about this particular trial is that it involves academics or researchers using sort of social media, so basically WhatsApp, to have real-time conversations with rice farmers to try and guide them through the actual process of what they need to do. I mean, if any of you have been involved in undergraduate teaching, I teach undergraduates in Sheffield, and it's amazing how undergraduates, if you give them a simple practical class, will still do something wrong to destroy or not replicate what you're expecting them to do. So what we're doing here is, or what Mashita's doing is, getting a normal rice farmer to replicate an experiment. So again, these experiments are still ongoing, but these are just some really sort of initial data. So again, this is from the field site in Borneo. The right-hand side is if you like the normal way of growing the rice plants, and then here on the left-hand side, you can see how they've been grown with this low density. And really just by eye, you can see they look very, very different from when you grow rice seedlings at a very, very high density. And if you look at the rice plants a bit closer up, then hopefully we can see here again, the image is pretty rubbish, but already at this very early stage, they've actually got quite a compact root ball, quite a sturdy root ball. And already hopefully you can see that there's a number of tillers already appearing at this very early stage. Whereas if you just go back a slide, clearly, if you were to take a similar picture of these seedlings here, there's no tillers happening. They're just going straight up because they're so packed together and haven't had the opportunity to, if you like, spread out. Again, these are really early data, so I wouldn't put too much emphasis on them. But again, from the early counts, it's very clear that these seedlings grown at low density have a much higher tiller number already at this very early stage of their growth. And so what we're doing now is to find out, we're letting those plants grow out, and then hopefully in a few months' time, we'll find out whether at maturity, that early increase of tiller number is translated in an increase in absolute tiller number of the mature plant, and does that also translate into actual change of yield of those plants. So again, this is compared to the first part of the project that we talked about about the e-leaf, about the modeling, which is much more longer term, understanding, modeling how plants work, and then hopefully identifying traits that can be brought into rice breeding processes as almost like an accident of observations of what we were doing. We may have identified elements which can lead into relatively straightforward or simple, hopefully simple changes in agronomy, which actually already might be able to help farmers. Okay, and again, this the idea of that the early establishments of seedlings is important. Actually, it chimes with work that I'm also doing in Sheffield, where we work with wheat breeders in Europe And again, one thing from my conversations there is that the early establishment of a seedling in the field is actually really, really important, that it sort of sets the scene for how well that plant will be able to resist disease or infection or stress later on in its lifetime. And again, I think lots of people working in crops know this, but it's probably not something that's been really highly emphasized. So again, that's something we're very keen to follow up on. So to summarize, as I said, this part of the talk has been relatively brief. Just want to give you some idea of the work that we've been doing in trying to take the basic fundamental science and translating it into something that could be of use to real life rice farmers. And I think there's two parts to it. First of all, this idea that you could actually use CO2 from mycelium to alter the early growth of rice, and that it might have economic value as a sustainable way of increasing rice yield. And then the final part, which you've just sort of again fallen into almost by accident because of, if you like, poor experimental design or doing something that you didn't really think about, but actually, if you look carefully, think actually this could be of use. So the idea that maybe the early growth of rice seedlings is really important, and that if we can increase the robustness of those rice seedlings, that could be actually very useful for rice farmers. Okay, so that's the end of the science part. I think we're almost at, I guess, a few minutes at the end, just to say a few words about reflections on international collaborations, to say the opportunities. Because I say this project has been running for a number of years, and it involves, I say, the main places in Shanghai, UPM, and Sheffield. And yeah, I don't know how many people here are involved in international collaborations or what the experiences are. First of all, I think they're great things to have. I think it's really fun. And again, this is one thing that actually, I know someone called Otlin Liza, who's the basically head of the research fund in the UK now, and she's very keen to sort of bring the element of fun back to science. That very often now science is so competitive, and there's so many pressures on academics and people working at universities that perhaps we've lost that element of enthusiasm for doing science because it's fun to do, and I totally agree with that. I think it's also really important, as one nice thing about this project is that I say I'm not a rice scientist by any stretch of the imagination. I'm not an agronomist. I'm actually much happier in a lab on a microscope. That's where I'm most comfortable. And then we have the guys in Shanghai who are world-class computational biologists. And again, I understand what they're doing, but the actual details of the coding, that's absolutely way beyond me. So they bring that speciality. And then the folks here in Malaysia, they really understand growing rice, so they bring the real world aspects on the challenges and opportunities there. So again, for all those collaborations, it's really important that all the partners feel that they can bring something to the table. I think during those collaborations with people from different countries, different cultures, you find there's different ways of, not different ways of doing science, but different goals of science. And again, we've had long term about publications, for example. So in the UK at the moment, we've just had the REF results, and in the UK, it's very clear that the goal now is in quality rather than quantity. So the pressures on me as a scientist are very much to get a relatively few, but really outstanding, excellent publications. In Malaysia, I think there's some pressure for number of publications and also good publications. And I'm also sure if Xing Wang were here, but also in China, there's also different pressures on academics, and understanding what those pressures are in different scientific communities, I think is really important for collaborations to work. I'm not saying we've got it perfectly right, but at least if you're aware of those issues, it's good. And just different ways of cultures of science between parts of Asia, parts of the US. And again, understanding those different ways of working is really important if you're going to have an effective way of working. And also just again, this is something that students often come, oh, I've got a bad result. There's no such thing as a bad result, unless, I mean, if it's a bad result if it's done badly, as in the experiment's rubbish and you can't interpret it. But results that don't fit with a theory or are not what you expect are actually good results, because that's how science progresses. There's no such thing as a bad result. And again, certainly in the UK, it's often a challenge for students to get into that mindset that they have a prediction. This is what the professor wants to see, and if I don't see it, it's bad. And that's absolutely not the case. And I think this is nicely shown by some of the data I've shown here, where we got results which weren't really what we expected. But actually, from those bad results, if you're aware, then you can actually sometimes turn them around to be positive results, because it leads into a line of research which is novel and actually advances science. So again, I think there are different views on what good science and bad science is, and you can get, it's very dangerous to get down a line of thought that this is the right result. This is the result that we must have, and that's a really dangerous way for science to go. So as long as the experiments are done well, you've got the appropriate controls, all the results are good. That's certainly my view. And then finally, the challenges. Obviously, we've had a pandemic, and again, really more to the more junior people in the audience, to worry too much. I'm sure a lot of you have found it really, really challenging. You've lost data, lost time. Certainly, I know that in UK, all senior academics are very, very aware of that. So if you're doing your PhD or whatever, the people who will be examining you will be very understanding if things haven't really worked out as one would have hoped over the last 2 years, because it's been tough. And actually, again, looking at UK, the PhD students in my group have been fantastic. They've adjusted their projects and got through. I'm sure that's true here. Funding, again, this is an eternal challenge for academics, I think, the world over. So I mentioned this project was funded by the GCRF, which is a great project, had very large sums of money, which enabled UK researchers to reach out and to collaborate across the globe. And then there was a change of government, and the funding just got turned off, which is really, really sad. I mean, so our project is quite lucky, but I certainly know there are other GCRF funded projects that essentially almost overnight just got turned off, and that's really, really tough to face. So again, that's part of, and I appreciate the pressures that governments are under. I mean, there's always a competition for funding and money, but yeah, those things can happen, and that's a tough part, especially when it involves working overseas with people who, it gets sometimes almost embarrassing that suddenly, oh my God, there's no more money. Sorry, that's a tough conversation to have. And then resilience, because again, there's pandemic challenges, there's funding challenges, and also just science itself is really challenging, and it's having that resilience to get through is important. And then the final point, I think, again, in this project and other projects I've been involved in, it's been really great that we've got some really talented early career researchers coming through, and it's true for this project and others. And I think that's one thing that, again, the international collaborations allow, is that it allows early career researchers to move around the globe, to visit other labs, see how other people do things, learn what's good, maybe learn what's not so good, and take that information back to their home countries and home universities, and to give those people a chance to develop and shine. And I think that, again, that's a really important thing to do at this moment in time. Okay, so that's enough from me. So since it's now 11:30, again, just to acknowledge the folk who have been involved in all the work that I've been talking about, and again, acknowledging the funders of the Royal Society for giving us the money to allow us to travel at last to see people. And again, I'm very happy to take questions on either this part of the talk or the previous one. So thank you for listening.
So we open the floor now for questions. I'm sorry.
Can you hear me?
Yes, I can.
So I'm wondering if your model is species sensitive, or is it a model just behave generally? Or if instant, if I want to see whether a Japonica behave like this or Indica like this?
That's a very good question. So yeah, it's all been based on Indica IR64, and again, we don't have. It'll be really part of the initial project was indeed to start to parameterize the model using data from other rice varieties. The amount of work involved in getting the data just for IR64 was pretty immense, and so we don't have data to back up that it's true for other rice varieties. We'd really love to do that. Obviously, our expectation is that it will work as a basis for most rice, but we'd love to do those experiments. It'd be good to have the funding to do that. So yeah, I can't say it will, but we're reasonably confident that it would be true for most rice varieties because the structure is comparable. Yeah, sure.
Is there any possibility that when you cultivate mycelium, the pathogen from mycelium can infect growth seedling in the mass scale or in the large scale?
Sorry, I didn't quite hear you.
Okay. Is there any possibility that the pathogen from mycelium, when you cultivate the mushroom, can travel to seedling and infect maybe?
I would hope not. And again, this all depends on I could pass it over to Armin, since he's the one who's actually doing the experiment. I suppose it depends on there's could be contamination in the lignocellulose, I suppose is what you're talking about. And hopefully you're seeding with the hyphae from the oyster mushroom. Hopefully that's relatively clean. But I don't know, Armin, have you ever seen any infection at all?
So what happens is the mycelium, the mycelium bags do get contamination from time to time, but we check the priming system regularly and remove any contaminated bags, because the bags that got contaminated, pretty much they are dead, and the mycelium cannot grow further and then result any to meaningful CO2 production. Okay. But towards the end of the mycelium growth, while conquering the mycelium bags, we do get lots of spores, and this is not really a problem. The only problem that we got is it's kind of sticking to the fan and kind of dampen the fan speed. Yeah. So this is one of the things that you need to bear in mind while doing it. The mycelium, the mushroom, it's from another kingdom. So that requires a different kind of knowledge to deal with it. Because rice, we know how to grow rice, that's the plant bit. Yeah. So contamination is not really a problem.
Okay, so it's not as long as you take care, I guess, is the answer.
Okay, is there any more? Ada soalan lagi ke? Ramai daripada agensi Paddy, ada apa-apa soalan berkaitan? Ada wakil nak tanya soalan? Ya, Dr. Norida.
Saya mewakilkan agensi lah bertanyakan soalannya.
Boleh.
Okay, this is related the field works about the CO2 priming of the seedlings. So in the field, sometimes the farmers are using the direct seeding. Direct seeding, forecasting. So how using this technique can help to increase to want to prime the
Yeah.
seedling on the field?
Yeah, I think basically not. So I mean, so this is very much focused on the communities that are using very traditional methods of rice growing, where you have it in a nursery and then transfer it. So for direct seeding, yeah, increasing CO2 in the field, in theory, you could do like face experiments, but that would be horrendously expensive. So I think our view is that this would be much more useful for, I think, in Borneo or in parts of India or parts of Philippines as well. There are sort of still parts of the world where people grow rice in a very traditional way, and this would help those folk. For direct seeding, then this is not going to be useful for that approach.
It was kind of accidents actually during the experiment. So we found out we wanted to test for the priming, but we found out something else. Okay, so and apparently the second thing that we found out later, it's actually useful for farmers. All right. Okay. You're good? Okay. Right. Okay. I think that's all from the audience.
Okay.
Right. So thank you very much for the kind words. Yeah. So it's been a great collaboration with Andy and the group and Jen. Okay, we have been working for very long now. I think I've known you for over 10 years now.
I think so. Yeah.
Yeah.
It's a long time.
Yeah, it's a very, very long time. But when you find people that you can work together for a long time, when it works very well. Yeah. All right. Okay. Is there any short dia nak cakap apa-apa kepada our collaborator? Sebab nak tutup lah. Dia nak cakap apa-apa? Say thank you to our visitor. Sebab dia nak balik atas ni.
Hello, my name is Nora Zura. I'm the Dean of Faculty of Agriculture. First of all, I have heard a lot of good things that have been said by Dr. Nazmin Amin, about this project. And I hope that maybe in future, if there's any, because he has found out new things about how the mycelium can help with the elevation of carbon dioxide. And I hope that later on, in future, if other agencies, I think that here can also, look this as much as an opportunity, because all this while we'll be looking at, having a breeding kind of work that might take some time also. But I think with the complement by this technique, agronomic practices, I think that also can increase the yield, which is a good thing. Yeah. So I hope that this project, by Nazmin and also the team here, and so from other institutes all around the world, can look into this and focus more. And I'm sure that we have the representative from the ministry here, Dr. Zulkifli, and also Mardi. Yeah. So I hope this can be looked further, and we can look how it can be, make it a bigger project. So thank you very much. Dr. Nazmin has been updating me from time to time, during the pandemic, how is the frustration of getting this work done. And the faculty really support this project. Thank you very much. And I hope you have a pleasant stay here. I think it's 2 weeks or 1 week?
Just 1 week. I think we fly back tonight.
Yeah, I think they have all last week in Peninsula. How about in Sabah? Yeah, okay. Thank you very much. And very, very excellent work that's been done. Thank you. Yeah.
Thank you.
Thank you. So I think that's all for now. So if you haven't got any more questions, so I think if you've got further questions that you maybe didn't think about it now, you can email me later or anybody in the group regarding the project. So we are happy to answer any questions about it. Okay. about rice, about anything about crop science in general. So thank you very much. Thank you for your time. So I'll see you again in the future.